Good morning, everybody. This is Mike Cavanaugh, a senior principal at Geosyntec Consultants in Oakland, California, and I'm going to be facilitating today's webinar in the SERDP and ESTCP webinar series. We have today an overview of the SERDP and ESTCP program first by Dr. Robin Neeson, and then we'll be followed by a list of some of the upcoming webinars in the series. After Dr. Neeson's remarks, we'll transition to the technical portion of the webinar. And today's event features two presentations highlighting advances in the assessment and in situ treatment of contaminated sediments. And I'm personally looking forward to this. I've worked mostly in the groundwater contamination area, but the sediment story is an important one. First, Mr. Gunther Rosen from the U.S. Navy's Space and Naval Warfare Systems Center will discuss the development and demonstration of an in situ toxicity and bioaccumulation testing technology. His presentation will be followed by a brief question and answer session. Second, we have Dr. Bart Chadwick, also from the same organization, who will talk about pilot scale performance of in situ treatment with reactive carbon amendments for contaminated sediments at an active DoD harbor site. We'll conclude the webinar with an interactive Q&A session with both of today's speakers. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions, and so we certainly encourage you to submit them during the session. On this slide, we've provided a few suggestions in the event that you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line. However, if you continue to have problems, please submit a comment using the chat box in the lower left-hand corner of your screen. With that, I'd like to introduce Robin Neeson, who will introduce the SERDP and ESTCP story. He's our the program manager for the weapon systems and platforms. Before joining SERDP and ESTCP, Robin worked at the Navar's Weapons Division in China Lake, California. So Robin, take it over.
Thank you, Mike. First of all, thank you all for joining us today for our webinar. Appreciate you all being here. And next slide, please. So I want to talk a little bit about what SERDP and ESTCP are. SERDP is the Strategic Environmental Research and Development Program. It was established by Congress in 1991, and it is a partnership of the DoD, DOE, and EPA. We're a requirements-driven program, and we try to identify high-priority environmental, science, and technology investment opportunities which address DoD requirements. We do both advanced technology development for near-term needs as well as fundamental research that impacts real-world environmental management. Next slide, please. ESTCP, on the other hand, is the Environmental Security Technology Certification Program. It's a demonstration validation program. We demonstrate innovative, cost-effective environmental and energy technologies, and we try to capitalize on our past investments from SERDP and elsewhere, and we try to transition those technologies out of the laboratory. We also intend to promote implementation and facilitate regulatory acceptance of these technologies. There are 5 program areas. First of all, energy and water is ESTCP only. Environmental restoration, munitions response, resource conservation and climate, that should be resilience, resource conservation and resilience, as well as weapon systems and platforms are all both SERDP and ESTCP efforts. Next slide, please. Today's webinar is focused on our environmental restoration program area, and within environmental restoration there are several focus areas, including contaminated groundwater, contaminants on ranges, contaminated sediments, wastewater treatment, and risk assessment. Next slide. And at this point I just want to talk a little about what's coming up. In the next couple of months, what we're doing is our award-winning projects. So each year we select from our SERDP and ESTCP projects that are completing, we select projects that are our projects of the year. And so in the next 2 or 3 months, we will be presenting several different award-winning projects, and I invite all of you to look at those on our website and consider participating in our webinars as they go forward. And with that, I'm going to pass it back to Mike to introduce our speakers.
Great. Thanks very much, Robin. So I'd like to take this opportunity to introduce Mr. Gunther Rosen. Gunther is an aquatic biologist with the U.S. Navy Space and Naval Warfare Systems Center in San Diego. And for the past 18 years, he's been conducting basic and applied research for the Navy, primarily in support of improved management of contaminated discharges and sediments. His research focuses on understanding the bioavailability and toxicity of DoD-relevant contaminants on marine biota and the development or demonstration and validation of technologies to improve environmental quality assessment. Mr. Rosen received a bachelor's degree in aquatic biology from the University of California, Santa Barbara, and a master's degree in aquaculture from Oregon State University. So, Gunther, take it over from here.
Thank you very much, Mike, for the introduction. And good day, everybody. Thanks for making it. This project that I'm going to be talking about today goes back a number of years, actually, starting with a SERDP effort that was led by Dr. Alan Burton from the University of Michigan, where we developed and tested a proof of concept prototype in situ bioassay device. Ultimately, it wound up getting funded under the ESTCP, where we had some pretty ambitious goals of developing a commercial prototype, demonstrating it at DoD sites, and then also facilitating regulatory acceptance. So I'll be talking about parts of that today. Just an overview of the talk. I'll discuss why certain DoD environmental challenges or issues can't really be adequately assessed using standard laboratory techniques. I'll discuss some of the advantages and disadvantages of in situ bioassays. This will be followed by a description of what we call the C-ring, the Sediment Ecotoxicity Assessment ring. I'll talk about its description and its development, followed by some highlights of performance at a couple of our sites, including Marine Corps-based Quantico, which was a thin layer cap monitoring site, and Naval-based San Diego, where we were up in the water column doing some unique stormwater monitoring. Finally, I'll talk a little bit about the technology transition progress we've made. So this cartoon here shows some DoD relevant kinds of situations where you really can't necessarily represent the exposure in a grab sample in the laboratory. So we've got a series of time-varying stressors here. For instance, groundwater seepage coming through the sediments, leakage of munitions constituents at former training ranges, stormwater and stormwater particles potentially depositing on sediments that have been remediated, resuspension of sediments from propeller wash. So those are examples of time-varying stressors. We also see a need for in situ types of assessment for these increasingly used in situ remedies, for instance, activated carbon, where you can't really take a sample and homogenize it and take it back to the laboratory. You really need to, in order to really understand its effectiveness, you need to keep the sample intact and conduct it on site. So a few of the advantages of conducting studies in situ are really just you're improving realism. And number one, integrating both spatial and temporal variability. I mentioned time-varying stressors, and then also preserving that maybe vertical stratification that you might have, for example, with the in situ remedies. You're also integrating natural factors, site-specific water conditions. You're minimizing sample disruption. And you can also wind up reducing costs. You don't need to collect samples to take back to the lab. You don't need to worry about holding times. Of course, like anything else, there are also disadvantages. You do lose an element of control when you place live organisms and chambers out in the field. you can wind up with one of the areas that I've really focused on in this project is trying to make sure that we can properly characterize water quality during the exposure. Some of the more traditional mesh type of designs have involved clogging, fouling, and then you can't really say for sure whether or not the water quality was acceptable for the organisms to be healthy. You can also have physical disturbances, toppling over of chambers, predation, some of your test organisms, if they're consumed by other organisms, that can be a problem if you're trying to enumerate them. You can have equipment loss, and then there's also tends to be a need for divers, which can add to expenses. So in this slide here, I'm just showing two basic designs. These are traditional, very well established designs that have been used for decades. The one on the left is Alan Burton's classic standard chamber. It's basically a core liner with a mesh cutout and two end caps. The one on the right is by Brian Anderson's group up in Granite Canyon, the Marine Pollution Studies Lab in Monterey. They've got a vertically oriented chamber that they experimented with an open bottom, so preventing any major disruption of the sediment. It's just pushed straight down, and organisms are also released through a syringe. So these two types of passive types of in situ chambers, they're inexpensive, and they generally do the job, but you do have this assumption that water quality is maintained, and these methods really aren't standardized. So our goal in the ESTCP project was to take the best of the traditional designs and try to develop a more standard test system that also increased the element of control so that we could actually potentially meet and document the types of test acceptability criteria that you would expect in a laboratory type facility. So the general design you can see is it's a circular carousel-shaped design. There's a center core that has the brain of the brain and the programming capability. It also includes a series of 10 pumps that feed individual chambers, and water comes in through this integrated chamber cap. You're looking on the bottom left picture, you're looking at the top down, and that integrated cap is where water is fed in from the pump. This is ambient water adjacent to the C-ring, and then it comes back out through another port in that same cap. So you don't need the mesh chamber anymore. You've got solid chambers for just ease of use and easy to replace. And we have reduced clogging and tendencies. And we can program our pump rate to whatever we like, and we can document and verify a data file was produced, and we can document and verify that the pumps worked properly or not. The other big thing for us was to make sure that we were using all non-toxic and cleanable components with as few consumables required as possible. So you can see here, just moving from left to right on the bottom, sort of the deployment kind of preparation, we're adding worms through a syringe. You can also include passive samplers, and then it gets deployed and recovered, and you're seeing here on the right a recovered C-ring from a sediment deployment. We typically try to push these into this base plate, which gives you about a 5-inch core. And here, just looking at a cross-section of the chambers and the different types of chambers that you can use depending on your study goals. You can conduct, in essence, like a toxicity identification evaluation of sorts by compartmentalizing the exposure. On the left, we have an overlying water exposure by capping the bottom of the chamber. We can do a sediment-water interface exposure by using a mesh bottom and potentially looking at the flux of contaminants coming up through into the water column. And then we can the more traditional sediment core exposure with an open bottom for our benthic invertebrates interacting with the sediment. We can also, as I mentioned, integrate passive sampling devices for multiple lines of evidence, looking at other bioavailability measures, and also integrate water quality sensors into the chambers as well to capture water quality inside the chamber, not just outside. Just a quick close-up of some typical components here. With the pre-filter removed, you can see the 10 pumps. These are easily replaceable and very inexpensive and low power. Some of the water quality sensors we typically use are trolls or hobo, conductivity loggers, and DO loggers, dissolved oxygen loggers. We've got a variety of sediment capture devices to recover the sediments and our test organisms. And then we've worked with several, by leveraging with several other projects, we've been able to integrate SPEMEs and DGTs and other types of passive samplers into our efforts. So with respect to regulatory acceptance, one mechanism we pursued was verification under EPA's Environmental Technology Verification Program. So this is a third-party validation that was orchestrated by EPA and Battelle. And it was a pretty rigorous laboratory-based study. So, of course, it's a little bit hard to compare to the standard when the standard is in the lab and you're going to get a different response than in the field. So the way this was dealt with was to have an apples-to-apples comparison by conducting all the testing in the laboratory, including the C-rings, just to verify that the C-ring would produce or not produce the same type of results that a standard laboratory test would. So this was a rigorous effort involving 5 species, both aqueous and sediment toxicity testing. 3 species were used for sediment bioaccumulation on several sediments. And just a quick snapshot here. You can download the report at the link down below here, but just a quick, but overall, we found very similar results. And here, this figure here shows for Macoma and Neanthes, a clam and a polychaete, PCB uptake in the C-ring compared to the laboratory was quite similar in a sediment sample collected from Puget Sound Naval Shipyard. Speaking of Puget Sound Naval Shipyard, we've been there 4 times, in doing we did a baseline assessment there to look at a site, Pier 7, where PCBs were the contaminant of concern, and then 3 post-remedy monitoring efforts. We used the Macoma again and Nephtes, Sequoides, polychaete, both of which are present in that area, and also integrated in situ SPEMEs. And this is just a teaser, really. Bart is going to be talking about that project in depth in the next talk. I'll be talking a little bit about another one of Bart's projects, which is based in Marine Corps base Quantico. This was, the remedy at this site was enhanced monitored natural recovery, which involved eventually the placement of a 6-inch sand cap on top of DDT contaminated sediment. And this was located in Quantico embayment, just adjacent to the Potomac River. You can see in the lower right, the green overlay was the anticipated sand cap placement footprint. This is very close to the shoreline where there were numerous sources now gone, an old landfill and a pesticide storage facility among some other potential sources that contaminated the site. So you can see here we have 5 stations where we placed C-rings on the cap, as well as a baseline before the cap was deployed, and then we have 2 off-site, off-cap stations as reference stations. 2 species were used. This is a freshwater site. We used Lumbriculus variegatus, a freshwater oligochaete, commonly used in bioaccumulation testing, and the Asian clam, Corbicula. The oligochaete is a deposit feeder. The clam is more of a suspension feeder, filtering particles at the sediment surface. And we also integrated, through leveraging with Bart's other project, ex situ passive sampling, and through Danny Reibel's assistance, some in situ passive sampling as well. So the purpose of this slide, these are results from our baseline assessment. And by the way, I want to mention that I'm really focusing here on anything sea ring related. A tremendous number of other measurements are being made at the site, but I don't have time to talk about those. So this is just a very focused effort here. So these are baseline results for the 2 different species. This is expressed as the sum of DDT and its metabolites normalized to lipid. On select stations where we did both laboratory and in situ. So we're trying to validate the utility and usefulness of the sea ring here. And one of the goals was to look at variability. And how are the results much more variable than they are in more controlled laboratory studies? And what we found here was that the trends were pretty similar overall in terms of contamination. Q1 on the left is the north part of the northern part of the site, which is historically the most contaminated. Q6 on the other end there is one of our reference stations. And you can see here, I don't have the standard deviations shown, but I've got the CVs, and our variability between the laboratory and in situ are very similar, both for Lumbriculus and for Corbicula. Another thing to note here is that Corbicula, the scale here, Corbicula bioaccumulated at about an order of magnitude lower concentrations as compared to the sediment deposit feeding worm, which you might expect. But both organisms worked well in the system, and variability was minimal. But the bottom line is in situ and ex situ results are indeed different, which is why we do these kinds of studies. This is also just a snapshot here. We are seeing reduced DDX bioavailability. We've got plotted here baseline concentrations for the oligochaete, and then 2 months post-cap installation and 14-month post-cap installation concentrations. This is on average of the 5 stations on the cap. So the sea ring here is placed on the sediment surface and penetrates about 5 inches down. Of course, if anything is moving up through the cap, it would be captured. But we are basically exposing our test organisms and our passive samplers to the cap. So we saw quite a nice reduction that seems to be sustained. And we also saw the same kind of result with the ex situ SPEME. So SPEME were rolled in the laboratory for the same period, the same exposure period, which was 14 days in this case. And we see a similar reduction in the pore water concentration as reported by the SPEME. The in situ SPEME results are also showed decreases, but not as significant. And again, here we're looking at the top 5 centimeters. Did our comparisons since the organisms are really living in that top 5 centimeter area, and we saw a nice strong correlation between the bioaccumulation and the ex situ SPEME. I wanted to show an example. I mentioned water quality and the need to really be able to assess that and count on it. And what you can see here, a hobo logger on the right, which is sitting right at the sediment-water interface, the sensor's at the interface there, and it is built into a modified C-ring chamber cap. So there's a seal there, and we're capturing actual water quality inside the system, not outside. We do place the sensors on the outside as well and make comparisons, and you can see in these pictures on the left, the top left, this is on the sand cap for one of our stations. The inside and the outside concentrations for dissolved oxygen are very similar and above any critical thresholds. Temperature, again, very similar as well. On the bottom, we're looking at our reference site, which is the sediment is much more fine, potentially has higher sediment oxygen demand, and we're doing pretty well here as well. Inside and outside conditions are the same. So moving on to a different application here at Naval Base San Diego. This is our backyard. For years, we've been watching our NPDES permits becoming increasingly stringent for stormwater compliance. And we've done a number of studies, which one by Katz et al., you can download this paper as well, or actually a report. It's one of the most exhaustive, intensive studies of stormwater toxicity testing available. We looked at 4 different Navy bases in the region over 3 years, first flush and composite collection methods at the end of pipe, and then also in the receiving water. And what we were seeing was frequently we'd be failing our NPDES permits for the toxicity requirement, but in the receiving water we never saw any toxicity, and this was over and over again. So we thought that using the C-ring as an integrative tool to assess these kinds of situations and provide lines of evidence for what the real impacts to the beneficial uses are would be a nice demonstration. So on the map here, this is Naval Base San Diego. The top red circle is Choyas Creek, an urban water draining from the urban watershed above, and then the smaller circle is one of our outfalls, and we set up C-rings in a gradient away from Choyas Creek, and then in a gradient away from the outfall down below as well. And we placed these at 1 meter depths and 3 meter depths. And threw in there several different species, mussel embryos, the polychaete anthes, mysid shrimp, and kelp for looking at kelp spore germination. We also integrated, in this case, DGTs for looking at labile metals because copper and zinc tend to be the most common contaminants of concern in stormwater in San Diego. So what we found from this study was very encouraging, and we were very pleased at our reference sites where we really wouldn't expect to see anything. All of our organisms performed well in the C-ring. Our greatest adverse effects, they were at Choyas Creek in the top near the surface, but that was driven by salinity. There were major dips in salinity to near zero several times during the first 48 hours of the storm, and these are marine species. So we don't believe we're looking at contaminant-related effects here. We're looking at just the natural physiological threshold being exceeded from salinity. And very little, a couple of small hits, but for the most part, not really much of any significant toxicity. Unlike grab samples tested in the laboratory where at the end of pipe where we saw significant toxicity. Also, I just wanted to point out for dissolved copper, just as an example, we did exceed the water quality criteria at most of our stations during the storm, not by much. We're looking in the range of 4 to 6 part per billion. And those samples were collected by grab sampling 8 different time points over a 24-hour period during a storm, which is very difficult to do and very intensive. And we found that a really nice relationship with the DGT placed at the C-ring in comparison to the composited grabs. So we thought that this integrative measure with the DGT was really exemplified here in this situation and can potentially be a cost saver from collecting such laborious types of samples. As far as tech transfer, the hardware is commercially available from ZebraTech. There's an operation manual that's available for the user to understand how it works. We teamed up with several folks, including Alan Burton, who owns C-Rings as well now, Chris Stransky from AMEC, who has been really integrating the C-Ring into his monitoring programs, such as his ASBS program here in California at Scripps. And he owns C-Ring, so there's a growing number of personnel that have the expertise. There's a growing number of units out there that are available to potentially use. We did pursue the EPA ETV successfully. We've been working with DoD collaborators at all of our sites. Bob Johnson, for instance, at Puget Sound Naval Shipyard, has been really good at bringing the RPMs and bringing other guests to see these technologies in action. So they've been getting a lot of exposure that way as well. So I just have a quick 1-minute video, and we'll see if it works here. So if it works, this is basically a deployment up at Puget Sound Naval Shipyard at Pier 7, where Bart's going to be talking about in a few minutes. It's a C-Ring deployment at probably one of our more difficult stations. There's a lot of shell hash there, and it's a little hard to install. But I wanted to give you guys an idea of what it looks like to deploy the instrumentation. So let's give it a go. And these are Navy divers required to deploy these at the site. Of course, if it's soft mud, these things just slip right in. We had quite a large amount of biology at the site. Here we're releasing our polychaetes through the syringe. That was done by the diver. It can be done remotely. When the pump is running, a blue light flashes. And here we're integrating some of Geosyntek's passive sampling devices. I mentioned a lot of biology. There's quite a bit going on down there. A lot of entertainment for the divers. So to conclude my part of the webinar here, I just wanted to restate the commercial availability and increasing use of the C-Ring. We're using it in a number of different applications. Again, in situ assessments can be more realistic, and in some cases are really the only means of accurately accurate exposures. When appropriately used, we can meet test acceptability criteria required of laboratory methods. And ultimately, we're looking at potentially reducing costs, not so much through the testing itself, but by making better decisions associated with cleanup and compliance, and not unnecessarily relying on very conservative assumptions. This is a partial list of some of the folks who've been critical to the success of this project. So I want to thank all of them, and for additional information, our report should be available on the SERDP website shortly. I'm not exactly sure when, though. It has been officially accepted, though.
Gunther, thank you very much for a really great presentation on an interesting diagnostic tool. I want to remind the audience to submit questions by using the chat box on the left-hand portion. We have received a number of questions for you, Gunther, and so I'm going to read them and let you respond, okay?
Yes, sir.
So the first one is, what costs are associated with using a C-ring compared to standard laboratory testing?
Right. A C-ring is the cost of a C-ring from ZebraTech, fully outfitted, is on the order of about $6,000. So we've been using C-rings typically at sites, usually in quantities of 6 to 10. So you can do the math from there on what it would cost to acquire the hardware. There's just a few consumables, you know, like the inner core liners, for example, that need to be replaced periodically. We did do a pretty extensive cost analysis that will be in our report. And just to be succinct here, using a 10-station kind of a study design, we found that the C-ring actually wound up, for bioaccumulation testing with 2 species, the test design wound up being about 10% less than in cost than a laboratory-based program. That's not always the case. In some scenarios, it was 10% more to do it in situ, but really not much different overall.
Okay, great, thank you. And a detailed question about the pump. It needs power, so what's the, how long can the C-ring be deployed without having to replace the pumps?
Right. Yeah, that's a great question, and something we've studied, spent a lot of time studying and testing the limits on. At this point in the current design, we can comfortably do a 28-day exposure. I'd like to think we could also, if required, if it was required for the project, we could do probably up to a 56-day exposure. These new pumps are really low power and much better than sort of an interim pumping system we had at one point where we really couldn't entertain that option. So 28 days minimum.
Okay.
And it really depends also on the on how much flushing you really want. If there are certain objectives of the study that require more flushing or less flushing of the chamber, that can have an impact on the duration.
All right, good. So this is a question from Jennifer Sutter at the Oregon DEQ. I'm reading her out just because it's a regulatory question coming from that side of the story. So her question is, does the 14-month increasing SPME data at Quantico suggest movement of pore water up through the 6-inch cap? Right.
That was what we were thinking might be the case. Data are trickling in from the 2-year or 22-month. We went back just this past September, and the data are coming in, and it's looking like it's holding. We were thinking it might be going up, but apparently, if I'm correct, the preliminary data I've seen from Geosyntec shows that it's looking pretty stable. But that would obviously be a potential explanation migrating through the cap.
Okay, great. And there's numerous questions for you, Gunther, so there's a lot of interest here. Are there limitations to what species and toxicological endpoints can be used in the sea rings?
We've used probably on the order of a dozen marine and freshwater species at this point. I'm sure Alan Burton and others in this world of in situ testing have used many others as well. The only thing I can think of really that would be a constraint or limitation is certain endpoints really wouldn't make sense. What comes to mind is like the sea urchin fertilization test, which you kind of need microscopes for, and it happens on the order of an hour as opposed to days. It just wouldn't make sense to place to do those types of studies in situ, to my knowledge. Also, if you've got particularly large organisms, we would probably have to modify the chamber design. The width, the diameter of the exposure chambers are 2 3/4 inches. So we tend to use smaller organisms.
Okay. A follow-up question along those lines: are there limitations to the exposure duration associated with the sea ring deployments?
Yeah, I think that more or less I may have already kind of addressed before, just with it's a balance between the battery life and maintaining water quality, or whatever the exchange rate you really want to occur. But I think comfortably we can do 28-day exposures, and likely quite a bit longer.
Okay, and another detail here: what is the range of water depth appropriate for the sea ring use?
The sea rings have been pressure tested to 100 feet by the manufacturer, and I'm pretty sure most of our sites are going to be at less than 100 feet. Ours certainly have been. And then on the other end, in terms of can it be used in the intertidal zone, the current design requires about 12 inches of clearance of water depth for the pumps to work, but we thought of ways that we could actually do some minor modifications so that we could actually let the system go dry. If at low tide, it could be fully exposed and we could still have the system work.
Okay. And I'm assuming divers are required for both recovery and deployment. Is that part of the standard operational procedures?
It seems to be at the moment, but we're constantly seeking ways to eliminate that. So that piece is a work in progress. There are a couple of aspects that just require some focused attention by humans. So.
Right. Good. Has the technology been used to look at remediation-induced contaminant mobilization?
Can you repeat that one? Sorry.
Sure. Has the technology been used to look at remediation-induced contaminant mobilization? In other words, the way I would translate it is, what if you're doing some dredging and you want to understand the impact that the dredging might have? At least that's my interpretation of that question.
Okay.
And this is the last one, by the way, so we won't torture you any further.
I'll just defer that one to Bart.
Yeah, well, we can, okay, thank you, Gunther. I think what we'll do is end this short Q&A session. Great questions coming from the audience, a lot more of them, which we don't have time to cover. So certainly they're welcome to contact you directly for further responses. But we'll have a chance at the end to come back to that, and you can think about that remediation-induced contaminant mobilization. All right, thank you very much. Let me at this point move to the second part of the webinar and introduce Dr. Bart Chadwick. He currently directs research and business development for the U.S. Navy's Energy and Environmental Systems Group within the U.S. Navy Space and Naval Weapons Systems Center in the Pacific. Dr. Chadwick has extensive experience in oceanography, engineering, technology development, measurement, and modeling of the marine environment. His research spans areas of environmental assessment, climate change vulnerability, and energy harvesting. Dr. Chadwick earned his BS degree in engineering from the University of California, Berkeley, and his PhD in oceanography from the Scripps Institution of Oceanography. So, a California lad. Bart?
Great, thanks very much, Mike, and thanks everyone for being here. I'm going to talk about a demonstration project, an ESTCP demonstration project that we are just finishing up to look at pilot scale performance of in situ treatment using reactive carbon amendments at a DoD harbor site. And this is really building on a foundation of work largely supported by SERDP, people like Dick Luthi and Upal Ghosh, who worked on developing these amendments. And there's been a number of pilot studies for these amendments that have shown real promise as a sort of a new dimension in our remediation quiver. But the focus of our work was that a lot of our sites where, for the Navy and DoD, where we have contamination, tend to be in active areas where we still have naval bases or shipyards or significant amounts of infrastructure and activity going on, and there really hadn't been a detailed demonstration done under those kinds of conditions. So this project really focused on seeing if these kind of remedies would be effective under those types of conditions. So, excuse me, by way of an outline, I'm just going to talk a little bit more about the objectives and the approach for the project, and then describe the amendment that we used and a little bit about the demonstration site. Then I'll give a little bit of information on a lab treatability study we did before going to the field, a description of the design and the installation, and then the post-installation performance monitoring, some analysis of costs that we developed based on the pilot scale testing, and then just wrap up with some key points and considerations that we're still looking at. So I mentioned this a little bit. The demonstration objective was really to look at both the validation of the placement, stability, and the performance of the reactive amendment in this active DoD harbor setting, with kind of a focus on placement into deeper water areas around where there's vessel traffic and near piers and structures, looking at the effectiveness in controlling bioavailability, specifically in this case for PCBs, and then assessing to the extent we could the physical stability and longevity of the amendment in the sediment, and also one of the key kind of lingering questions, which is the response of the benthic community to these kind of amendment applications. So an overview, the project kind of got going in around 2011 with this laboratory treatability study that I'll talk about. Based on that showing good results, we went on to designing kind of the engineering design of the installation and the baseline monitoring, which happened in the summer of 2012. The installation happened in the fall of 2012, and then we had a series of subsequent monitoring events that went on for about 3 years, and then a slew of data analysis, and we're just wrapping up the final report, which should be getting done in the next few weeks and posting up to the ESTCP website. So the carbon amendment that we're using is a product called AquaGate plus PAC. It's made by the company AquaBlock. And the amendment is essentially a coated aggregate, and the coating is is about a 10% bentonite binder that holds on about 5% activated carbon. So it's basically a coating on these small aggregate particles. And the idea is that basically the aggregate aids in delivery of the carbon to the sediment surface, penetrating in a bit into the bottom as it settles. And then after a pretty short time, the bentonite starts to expand and it flakes off into the sediment and then gets vertically mixed over time just by natural processes at the site. So you can see there's a little picture here of the aggregate and what it looks like, and then on the right there's sort of a cartoon series of how we envision this working, and the carbon eventually getting mixed down into the surface sediment and interacting with the contaminants in a way that it binds them up and reduces their bioavailability. The demonstration site that we worked at is an area called Pier 7 at the Puget Sound Naval Shipyard. It's located in Bremerton, Washington, on the Puget Sound. And this is an active deepwater shipyard area. There was a previous remedial action at the site in this area they call the Operable Unit B. And a lot of the focus on that was for PCBs in surface sediments that were predominantly on the shoreline areas in amongst these pier areas that you see in the photograph at the right. And there was a combined remedy that included some dredging and then also some aquatic disposal and capping and some natural recovery. So during post-remedy monitoring, there were some residual areas identified in this area around Pier 7 where there were still some elevated levels, and so sort of created an opportunity to come in with this pilot scale test and see if it would be effective, and particularly under this kind of shipyard setting. So you can see the second picture on the right, on the lower side, kind of the activities going on, moving submarines around with tugboats and things like that, and these all have an influence on the kind of physical stability of the sediment and subsequently the amendment. So our first step was this lab treatability study, and what we did was we went out to the site and delineated that area around Pier 7 a little better and found PCBs kind of in the range of about 500 ppb to up to as high as 6 ppm in this kind of focused area right along the end of the pier there and off to the left into the berthing area. We collected sediments from those, brought them back to the lab, and did applications of the AquaGate in the laboratory using basically 3 treatments, one where we just added it on top and then did exposures, one where we mixed it for 24 hours, and one where we mixed it for a month. And you can see on the right the results from that basically show that we did achieve significant reductions in PCB uptake into the polychaetes, and it got better and better essentially the more you mixed it. This is sort of a well-known characteristic. So that's the dependency on this mixing property for the PCBs essentially to come into contact with the activated carbon. We also did some toxicity testing and didn't see any toxicity associated with adding the amendment. And we also used this as an opportunity to test the sediment profile imaging camera to see if we could visually see the amendment once it was placed on the sediments, and that was also pretty successful. So that was one of the key things we used to sort of track the amendment over time when we went to the field. So the design for the pilot scale application then ended up being about a ½ acre, and you can see in the figure it's an area it's about 115 by 190 feet long, and about almost half of it is underneath the pier. A portion of it is on the slope that goes down into the berthing area, then a portion of it is in the berthing area itself, shown by that that red dotted line. And then the design called for increasing the carbon content of the sediment by about 2 to 4% with activated carbon. That required about 141 tons of amendment, most of the weight of which is the aggregate. And that leads to an application of about a thickness of about 5 centimeters when the thing starts, and then an assumption of 10 to 20 centimeters of vertical mixing over time, and that drives that 2 to 4% increase requirement. So this slide shows some pictures of the application, and the first thing you'll notice is it's nighttime, and it wasn't just because we wanted to sneak in there at night while nobody was looking, but actually because to get underneath the pier areas required a really low tide, and during this time of year, the low tides were in the middle of the night. So, so you can see the application is basically on a barge. We had these super sacks that were fed into a hopper and then over a conveyor belt to this thing called a telebelt that was manipulated by a guy with like a kind of a joystick thing, and he could poke that thing around and swing it back and forth. So out in the berthing area, they would just basically swing it back and forth and telescope it in and out, and they could cover quite an area and just keep feeding it in. Under the piers, you can see that picture in the bottom right. It's a little trickier. You've got to get in between the pilings. They had to have a spreader plate that would try to get it in and around things. So, but they were really quite skillful at doing this, and I think did quite an elegant job in making all that work. So this next thing is a little movie that shows the installation. This just gives you a little better feel for kind of what it looks like when they're putting it in. You'll see some photos of the installation, and then some movies of the amendment actually coming down through the water column. You get sort of sense what that looks like, and then a diver transect as they swim along it. So I'll just let this play, and you can kind of see all this unfold. So this is the telebelt getting put under the pier to do the under pier installation. There's the transfer of one of the super sacks over to the hopper to feed the, and they whack it with a hammer a couple times to make it fall out. Now it's out in the berthing area, just the back and forth kind of installation. And then this was when the Navy divers actually went and sat on the bottom while we poured a little on top of them so we could see what that looked like. So you can see it raining down and hitting the bottom here. And then there's this diver transect. So they're going to start outside of the footprint and swim into the footprint. And then they end up over by the pier. Kind of you'll see one of the pier pilings. Again, like Gunther said, there's a ton of life down on the bottom here, especially crabs. So now you can see the aggregate is already, the carbon has already started to come off, and a lot of those white flecks are the aggregate. And the flounders have already matched their colors to the new amendment colored bottom. Anyway, so that's just mostly for entertainment value there. Let's see if I can get to the. Okay, so after the installation, then. The performance monitoring design basically had a setup where we did baseline before the installation, and then a series of post-placement monitoring surveys, a couple of which were mostly just to verify the placement, and then a bunch that were to verify the performance. And so the comparisons for the performance are essentially comparing the baseline condition prior to the amendment to the post-installation monitoring events. And there were 10 what we called multi-metric stations that were located within the footprint area. And then we had about 40 to 50 spy camera survey locations that were used to map out the location of where we could see amendment. And then we also had some reference areas. At the multimetric stations, we included everything, and then we had some reference areas where we used for the benthic community sampling. The methods we use, so thankfully Gunther just described all the C-ring work. So the primary tool we were using were C-rings, and so the two organisms, Macoma, the clam, and Neptes, the worm, and then that was integrated with solid phase microextraction, passive samplers for pore water measurements. Those were all done in situ. We also collected cores to look at the carbon and also benthic community analysis. And then the SPY survey, which if you're not familiar with, is essentially a camera with kind of a wedge-shaped front end that gets driven into the sediment and takes kind of a cross-sectional picture that you can use to analyze and look at both the presence of the amendment and also the condition of the sediments. So I'll walk you through some of the results, and just starting with the post-placement verification. So this is a map that's based on the spy camera work, showing areas of where the carbon ended up relative to kind of this red box target. So you can see essentially most of the area got treated. Some was a little thinner, and then down on the south end, those stations were off the edge. So there's some uncertainty about just how far south it went from this survey. And there's some areas outside the target where we found traces of the amendment too. So this is sort of our starting condition, and from this we assessed that about 80% of the area received at least a trace, and 70% received the target thickness of about 5 centimeters. And the average thickness within the target area was around 11 centimeters with a range of 0.1 to 17. And so this 11 centimeters seems, higher than the 5 target, but that includes some, penetration into the sediment already and things like that. So one thing I wanted to mention in the context before I show some of the subsequent data is the primary driver for physical disturbance at the site is believed to be ship movements, and particularly tugboat, as they push different ships and submarines and things like that around at the piers. So there was a leveraged effort by Dr. P.F. Wong, who at the same time was doing this evaluation of prop wash effects. And from data that he collected and modeling simulations that he did, which are shown kind of over at the right here, they estimate erosion on the order of about 20,000 kilograms for a typical kind of ship movement event, with a scour area that's typically on the order of 10 to 20 meters wide and maybe 50 to 150 meters long, potentially penetrating 1 to 5 centimeters. So this is the kind of thing that happens, typically like 4 events at the Pier 7 area per year. And one of the things we wondered is, if we would still see adequate performance with this kind of thing going on. So the next slide here then shows their simulation of how much stuff gets transported around following one of these resuspension events based on hydrodynamic modeling. So the figure at the top there on the right shows the resuspension of silt associated with one of these resuspension events and its transport by the tides. The same thing for sand, and then total. So you can see, relative to the scale of our site, which is fairly small, some of this transport is pretty significant. So so this is sort of the environment in which we're operating in. So now here, what I'm showing then is the 3 subsequent mapping efforts for the amendment that happened 10, 21, and 33 months post-installation. And what you see here is that the amendment, was still detectable through all the way out to the 33-month survey. And there are lateral variations in time, but we still see typically in the range of 70 to 80% of the area we were seeing detectable amendment in those areas. The average thickness stayed in that range of about 10 to 11 centimeters in terms of what we could detectably see with the spy camera. The uncertainties and the variations associated with this, the physical disturbance, I kind of showed you the potential for that. So that's one of the factors that we think is playing into this. The others are that there's this spatial variation, and we don't go to exactly the same station each time with the spy camera. It's just not possible. And so there's small scale variations that feed into this. And then the other thing is that over time, it just gets harder and harder to see the amendment as it gets kind of blended in. And so certainly in the later surveys, these numbers become, more and more uncertain. We did also do, as part of the coring, direct measurements of total organic carbon as an indicator of changes associated with the addition of the amendment. And so what you see in this figure are 3 sets of bars, and they're associated with different depth intervals into the sediment, 0 to 5, 5 to 10, 10 to 15, so getting deeper and deeper as you move to the right. And then the individual bars within each group are the different events, starting with the baseline condition and then all the subsequent valuations. And the first thing you see is there's quite a bit of variability in this. And then the second thing you see, the general trend, is that in the shallow 0 to 5 interval, the TOC went up quite a bit, especially in the earlier events, whereas as you go deeper into the sediment, you didn't really see it as much in the earlier events, but it starts to go up more in the later events. So this is, at least in the big picture, kind of what we had hoped and expected to see, is that if you lay down the carbon, initially it's fairly high in the upper intervals, and then over time we see it working its way down. And the deltas are kind of in that 2 to 4% range that we were targeting, although a lot of this is just lost in variability. So hard to say things really quantitatively, but at least qualitatively, this is in line with our expectations. And we also looked at black carbon content, which is in theory a more direct measure of the carbon, but we also know at some of these sites there's a fair amount of black carbon present even without the amendment. So, but these results, and they're formatted just the same way. They showed really quite a similar pattern with the higher concentrations in the shallow intervals early and then moving deeper. One kind of notable exception here was the 33-month event, where the black carbon seemed to be lower everywhere. We don't really have an explanation for that at this point, but could be aging, could be just inadequacies in our measurement or differences spatially and things like that. But the general trend, again, is sort of in line with expectation and gives us at least a sense that some carbon is hanging around and doing what we expect. So the direct measurements that we had for performance of the amendment were basically the pore water concentrations and the uptake in directly measured in the organisms on the sea rings. So I'm going to start off, and we're going to show you the pore water results. And so these plots are all going to look kind of the same. There's the baseline event on the left, and then the subsequent bars are the 10, 21, and 33-month events, and they're basically just showing kind of the these box and whisker type plots of how things trended. So what we found with pore water measured by these solid phase micro extraction fibers was reductions that showed up as early as the 10-month event and persisted through the 33-month event, and the reductions were consistently in this range of about 75 to 85%. So really pretty substantial. Again, you see there is variability from location to location, but the overall picture is a picture of substantial reductions, and in line with our expectations from the laboratory treatability study. Now this slide shows the results for the two different organisms that we use for bioaccumulation and sort of more real direct measurements of bioavailability. On the left is Neanthes, the worm, and here the same plot, you can see there's sort of an onward going downward trend, but reductions were here in the rate of 87 to 97% relative to baseline. And for Macoma, the clam, not quite as much, but reductions in the range of 68 to 88%. And again, things still going down in the 33-month event, at least in kind of a qualitative sense. So these results were really, consistent with what we saw in the pore water, and again, sort of substantiated what we saw in the treatability studies. So the final kind of performance metric we looked at was benthic community health, and so here I'm showing plots again of the baseline condition compared to post-installation, and then there's a second set of bars to the right that are for the control unamended areas that we tracked through the whole study for benthic community. So what we found with this was essentially that the benthic community metrics were comparable to the baseline and reference for a number of metrics, including abundance, evenness, and dominance. And I'm showing the abundance plot over on the right there at the top. And then some metrics showed changes from baseline, but they were still comparable to the reference areas. So for instance, diversity and taxa richness fell kind of into this class. So if you look at the plot on the bottom right for diversity, and you look at the amended stations, you see that there's some dropping off, say, in the 33-month event where the diversity appears to drop somewhat. But that diversity measurement was still consistent with the kind of range and response we were seeing in the unamended stations in that same area. So, so in general, what we found from the benthic community was that we couldn't really distinguish any significant differences in the field relative to what was going on in the general area. I'll just note that the blue dashed line on here is another independent measurement from the Puget Sound ambient monitoring program in the same general vicinity. So based on this, we took some of the data that was collected during the demonstration and used it to look at some hypothetical cost scenarios. And these were broken down into sort of different scenario type sites. Site 1 being somewhat larger, 5 acres, and assumed to have deeper, more complex kind of harbor setting with debris and infrastructure and the disposal having to go to hazardous waste. Site 2 was more medium-sized and maybe having slopes and still having upland but non-hazardous disposal. So, and then Site 3 is a smaller site, quiescent, depositional. So these are kind of going from more challenging to more smaller and simpler, kind of on the spectrum of remediation. So, and we just did cost estimates using the data we kind of developed during our assessment. And the main drivers for that are the material itself, the shipment and the storage, the staging and the placement, and then of course the monitoring. And what we found was that in cases of where you have large dredging volumes and refuse requiring removal and a lot of infrastructure to work around or potentially not even be able to dredge around, that the amendments seemed more cost effective. At other sites, as you go to these simpler trends, the costs appeared to be pretty comparable as well to things like capping. And so the bottom line here is that I think that it's all site dependent and everyone has to make their own decisions. But at least in a general sense, along with performing technically, it seems like these amendments can be pretty cost competitive. So just to wrap up, some of the key points. We do think activated carbon can be an effective remedy in these active harbor settings. We were able to demonstrate, an effective placement technique, even in kind of challenging areas under piers and on slopes. We saw sufficient stability and mixing of carbon in the surface sediment to achieve significant reductions in PCBs and tissue and pore water. We saw the remedy performance persist for up to about 3 years, and we couldn't really detect any significant adverse effects to the benthic community relative to other areas in the vicinity. And finally, as I mentioned, it does appear that, depending on site-specific conditions, that it can be a pretty cost-competitive remedy as well. And then I just want to finish up with a few final considerations. It's certainly, this is all new, and there's lots of questions and considerations I think still out there. But a few that we've thought about regarding the design and the placement. That one thing to think about in these active areas is the timing of the placement relative to when ship movements happen. So, for example, in our case, where we have 4 scheduled ship movements in that area, and they know when they're going to happen typically, that you could do the placement, just after one of them, and so the amendment would have some time to be there before it was disturbed. Another consideration would be the scale of the remedy, say, relative to some kind of transport scale. So I was showing those maps from the ship resuspension, and so if your site is very small relative to the distance that sediment is moving around at your site, that's a consideration for how, just how much dispersion there's going to happen with the amendment and how effective it's going to be right in the specific area where you're placing it. Which leads to kind of a final consideration that maybe with these and in these kinds of sites that we need to think about working with those kinds of processes versus trying to engineer against them and just design our sites with the expectation that things are going to move, and that is part of the understanding. With respect to monitoring, particularly for PCBs and other hydrophobic organic contaminants, a lot of the risk assessment is based on area-weighted strategies, and so coming up with area-weighted strategies to do the monitoring for amended sites is something that needs, I think, more consideration. Another key issue is for long-term monitoring, and this played into some of our cost estimates, is how long do you have to keep monitoring? If you've demonstrated it for 3 years or 5 years, or do you have to come back in 10 years? I think those are questions that still need some further discussion. And that's kind of key in this performance thing, where the long-term effectiveness greater than 5 years, I mean, there aren't a lot of sites that have had carbon out for time periods longer than that. So some of that is still sort of an open question. We only really demonstrated for PCBs. It's not an amendment or a treatment that will work for a broad spectrum of metals and all kinds of contaminants that you might have at a mixed contaminant type site. But there is a consideration for resilience, and one of the kind of chief advantages is that these amendments might contribute to the resilience of the site in terms of if there are ongoing inputs and you have some buffering capacity then with the amendment to still sorb and reduce the bioavailability of those incoming sources as well. And then just finally, from a regulatory standpoint, integrating these measures of reduced bioavailability metrics into remedial goals is something that's, I think, kind of, ongoing, and just figuring out how that works versus sort of typical bulk concentrations or carbon normalized concentrations and things like that. So I'm going to wrap up there, just acknowledging this is just a small fraction of the people that worked on it, and just a really great team, phenomenal effort, really, many challenges that were overcome. And I want to really emphasize the contributions that Vicki Kurtai made to this. She basically carried the load on this for a lot of the field work and making everything happen. So especially want to thank her for all her contributions. And more information on the website. As I said, the report I think will be posted hopefully in the next few weeks. We're just finishing up a final round of comment responses. I'll wrap up there, Mike.
Okay, Bart, thanks very much. Great talk and very interesting topic. We have a number of questions, but we've got limited time, unfortunately. 10 minutes left on the call. There have been some specific questions, and I want to ask the questioners to direct some of those detailed questions directly to you, Bart, if that's okay. Absolutely. I would like to raise a general question that comes up with the use of GAC amendments, and that's basically the long-term stability of the technology in different energy environments. I'm wondering if you could maybe give a little bit of an overview of that issue, because this technology's certainly been around for a while, and there's a limited amount of information over time as to its stability over longer time frames. How do you address that concern that would be posed by the regulatory side of the analysis?
Right. Yeah. Well, I hope I was reasonably clear. I think that there's been obviously a large number of pilot studies that have gone on, and there's more going on now. And I know SERDP-ESTCP has a project as well to go back and look at the long-term performance of some of the sites that they did early on in their studies. So I think everybody is kind of focused on that question and trying to get to the answer of it. I don't know that we're totally there yet, but I think part of what we learned from this particular demonstration is that even in a relatively, high energy or at least disturbed type of setting, the amendment seems to perform pretty well over a period of 3 years or so. I don't, we just don't have the data to go, out to 5-10 years and say more about it at this point. So those kind of considerations, I think, have to kind of get worked in, and whether or not you would have to come back and retreat, or if the thing is just going to be taken care of indefinitely. I think those are, some of that is going to be site-specific, and some of it is going to have to be answered by longer-term studies.
And along those same lines, is there any issue with respect to the capacity of the carbon, since it's an equilibrium process? What happens if you reach capacity for the PCB and it's not, for this particular chemical? Do you see that as an issue? That, in other words, the capacity of the carbon to address the contaminant of interest?
I think generally speaking, the capacity hasn't been an issue. The issue tends to be getting the amendment in contact with the contamination. So you can kind of see both in the controlled mixing experiments and in the performance in the field, we tend to see improvements with more mixing and with more time. And so that suggests that it's mostly a function of contact, not capacity. And there have been, work on different dosing rates, and, one of the issues is if you go to high dosing rates, you do start to see effects on some of the benthic community. So, these kind of, 2, 3, 4% ranges are kind of, I think, where people are landing to some degree in order to be effective, and also to, minimize any potential impact to, to the benthic community.
Okay, and one last question regarding the amendment. Is there any reason why you have to use a proprietary material like AquaBlock? Can you use generic granular activated carbon?
So, yeah, I mean, I think all of those kinds of materials have had some level of demonstration and showed levels of effectiveness. The carbon tends to be more effective, in smaller particle sizes in terms of absorption. So there's advantages to making it smaller in terms of its effectiveness, but that also makes it more difficult to deliver. That's why you end up with maybe a different kind of delivery system, like AquaGate or Sediments or something like that. And so those are kind of the considerations I think that people are balancing when they're making those decisions. And for our application here, we selected the AquaGate because we felt like it would give us penetration into the sediment and a better chance of having the carbon stay in place, when subject to some of these physical disturbances.
Okay, great. Well, thank you. We have a couple of minutes for joint questions. So from the audience, and let me read one of them. In what way do you expect the bioavailability from reactive amendments to change during using the different monitoring approaches in situ as compared to the lab? I guess this goes to the monitoring question, so maybe that's for you, Gunther, and then a follow-up.
Well, I think that Bart's slide on the treatability study addresses that pretty nicely. If you've got enhanced mixing, you're going to have more reduction in bioavailability. And so by modifying the approach to a lab-based approach, unless you are preserving that intact core and conducting your study like that, you're going to wind up having maybe an increased and potentially false sense of the value added. So what am I trying to? Sorry. Basically, that's how we would recommend. If you're going to do the study in the lab, you need to keep the sample intact. If you're going to composite it, mix it, and homogenize it, you're going to get false results for how it's actually working in the environment. So I think that would be our recommendation and thoughts, and I think that treatability study really exemplifies that response.
Okay, well, good. Bart, did you want to have the final word on that? Because we're running close to the end here, and just a final thought on that particular question.
Yeah, just one, I think, thing. So this kind of comes back to the area-weighted approaches and things like that. So, I mean, one of the things we'd like to be able to do is, collect, area-weighted samples or things like that and take them to the lab and look at the performance. And so, consideration for how to do that without doing a lot of mixing in the lab, where you're going to change the contact between the carbon and the contaminant, is sort of a key to that. And so people are adopting strategies like putting out multiple passive samplers and then compositing them, so you get the right exposure and then you composite once you've done the exposure, rather than changing the characteristic of the interaction of the amendment with the sediment.
Okay, well, again, I encourage everybody to contact you and Gunther directly. We need to go to the last slide here, which is the report on the next webinar, January 12th, on the award-winning projects for energy and water. And please take the opportunity to fill out the survey reminder. And with that, I will close by thanking everybody to have participated, and especially to our two excellent speakers. Thanks, everybody.
Thanks, Mike. Thanks, everybody.
Thank you.